Now that we understand the involvement of certain ions, such as sodium, potassium, and calcium, and others, and energy molecules, such as ATP, that are involved in contraction, let's take a look at what happens next. Heat is released from the process of muscular contraction. And we know this because some of the energy that is required to initiate work is lost to the external environment as heat. Recall that as energy goes from one state to another, you lose some of it in the form of heat. And this brings up the concept of efficiency. Efficiency is simply the amount of resourcefulness while accomplishing a task. And you may also want to note that the conversion of chemical energy into mechanical energy is not an efficient task, since only about 20 to 25% of the chemical energy is converted into the mechanical energy that is required to accomplish the job. In this context, if we want to look at the big picture, it is more efficient to use the escalator than it is to take the stairs. However, if the primary interest is in the efficiency of the actin-myosin cross bridges, then the metabolic expenses along with the basal metabolism and the excitation-contraction coupling that Celeste talked about earlier, together with the metabolic recovery process, must be all subtracted from the total heat and work observed. So, the last time you became overheated from a brief workout, you were experiencing a summation of all of the broken ATP bonds for every muscle utilized. And as I go through this video, it'll be helpful to keep in mind the work performed by smooth muscles as well, because even though they use less ATP, they still contribute to the overhaul of the muscular heat production in our body. One of the scientists who pioneered the earlier experiments on this concept was Wallace O. Finn. In 1922, Finn set out to find the relationship between work performance and heat production using a frog's sartorius muscle as a model system. Though Finn's experiments didn't draw any definitive conclusion about the relationship between the heat production and the muscular contraction, he led us into further investigation as we discover more about the mechanism of contraction. This increase in enthalpy that we experience is actually due to the breakdown of ATP into ADP plus an inorganic phosphate during muscular contraction. The harder we strain our muscles, the more ATP our muscle cells consume. However, during easier tasks, such as walking a dog, a really good dog, or even opening a door, uses less ATP than, say, running a marathon or even lifting weights. So now, as you may want to know, how do we attain this ATP? And what is the mechanism at which it's consumed? This process is called cellular respiration. You may remember that we have two types of respiration, anaerobic and aerobic respiration. Just as a reminder, you know that the anaerobic respiration occurs in the absence of oxygen and the aerobic respiration occurs in the presence of oxygen. As humans, we produce and store our energy from the foods we eat. Essentially, most everything we eat is converted to glucose. Now that we know we have a glucose molecule, we can create approximately 34 ATPs, but only in the presence of oxygen during aerobic respiration. If you recall, this process is the process of respiration. So remember that there are three cycles to aerobic respiration. There's the glycolysis, the Krebs cycle, and the electron transport chain. As a brief overview, two ATP molecules are synthesized during glycolysis, two ATP molecules are synthesized during the Krebs cycle, and 34 ATP molecules are synthesized during the electron transport chain. Again, that's only in the presence of oxygen. This is how our bodies create the ATP, energy needed for all of our bodily functions, especially muscular movement. So what happens when you run out of readily available oxygen or ATP during an aerobic workout? The short answer is that your muscle cells enter anaerobic respiration, which is also called fermentation. When your muscle cells run out of oxygen, glycolysis enters fermentation, which is your muscle's way of producing as much energy as it can without the presence of oxygen. The byproduct of our muscles' fermentation is lactic acid, which is why we feel sore after the intense workout. Well, when your muscles become deficient of ATP, they tap into stored polymers of glucose called glycogen, and a separate first aid attempt to provide the muscles with enough energy, and these processes can occur simultaneously. The body's usage of glycogen in this type of emergency also results in lactic acid because the glycogen simply enters the same cycle that glucose was in. This too produces lactic acid. And the bottom line is that acid hurts. And this is why we also feel sore after that intense workout. This results in muscle fatigue. Muscle fatigue is basically a decreased ability in a muscle cell's process of contraction. So, if you're like this serious workout buff and you're crazed about building mass muscle, you probably heard about an organic acid called creatine. And creatine has a reputation in the athletic world for increased energy production, and this allows athletes to train harder. But what exactly does it do? So, if we gaze at the schematic to the left, we'll notice that let's start backwards and then move our way back to the front. So we have creatine, and creatine is stored in our skeletal muscle. And with the assistance of a creatine kinase, we know that kinases phosphorylate molecules. So the creatine kinase phosphorylates creatine. Once creatine is phosphorylated, it becomes this phosphocreatine, which is the PCR. Now phosphocreatine is ready to add a phosphate group onto the adenosine diphosphate. Once the phosphate group onto adenosine diphosphate, we now know that this molecule is now ATP. So ATP is readily available for muscle contraction. You remember when Sunitha was talking about how the ATP lies at the myosin cross bridge head? So we were able to actually convert ADP right back into ATP at the myosin cross bridge without having to send that ADP back into the oxidative phosphorylation cycle. This allows for rapid muscle contraction without burning yourself out. The phosphocreatine, just before I elaborate on the oxidative phosphorylation, the phosphocreatine is an energy storage that serves to replenish ADP with the extra inorganic phosphate without having to send that ADP back into the oxidative phosphorylation cycle. And so now I want to kind of like go right back into the oxidative phosphorylation by describing it, kind of giving you a reminder that it's a part of the electron transport chain. And it results in the formulation of a hydrogen gradient, which will inevitably attach an inorganic phosphate onto the ADP. This process involves many electron carriers, such as an NADH, a FADH2, and a cytochrome C, just to name a few. It also involves four protein, that's actually four, I didn't mean to say 14. It actually involves four protein complexes and an ATP synthase. So as you may see, skipping that oxidative phosphorylation cycle to resynthesize ATP from an ADP simply by using creatine, that allows the muscle cells more time to build. So creatine is a very promising molecule in the area of medicine. Now that you understand the mechanisms of creatine and the muscular system, you may be able to understand its benefits in patients with muscular dystrophy. Other muscular disorders, as well as other areas in the body that have high energy demands, may also benefit from the usage of creatine. These include, but are not limited to, depression, intelligence, and memory. It is important to note that our body synthesizes creatine naturally, and this process occurs in the liver and is mostly stored in skeletal muscle. When working out or just living daily, we shouldn't be concerned with only building muscle from our skeletal muscle, because as you may know, smooth muscle and cardiac muscle are equally as important for optimal health. Since we now know about the mechanism of muscle, I wish to close by describing the categories of muscle fibers. There are two categories of muscle fibers. There's red muscle and white muscle. Red muscle is a slow twitch type 1 fiber. These muscles have small diameters, and an example of red muscle is the gastrocnemius muscle, which you'll see later in Zach's presentation. These muscles tend to be oxidative, so they use oxidative phosphorylation, and thus they have many more mitochondria than white muscle. Red muscle receives its color from myoglobin, and remember, that myoglobin is a protein that contains a heme, which is known to store and carry oxygen to muscle cells. I just kind of wanted to give like an example of like everyday red muscle that we see. So if you like to eat chicken, or the chicken's like our country's favorite meat, um, there's white meat and then there's dark meat. So the dark meat would be an example of red muscle, and the white meat would be a great example of the white muscle. White muscle is a fast twitch type 2 fiber. These muscles have a larger diameter than red muscle, and thus they're more powerful than red muscle. And an easy way to think of this is that the word twitch is referring to contraction. And I also kind of like to think of a helpful way to remember the different diameters of red muscle versus white muscle with this analogy. So imagine that you have to lift a heavy book, right? But you only have a few threads made of flimsy material to do this with. If you were to use one string, you may want to lift it very slowly because otherwise your string will snap, right? So the thinner the diameter, the slower the muscle. This is analogous to the slow twitch red muscle. And on the other hand, if you needed to lift that book pretty fast, that same heavy book pretty fast, you may want to increase the diameter of the string by adding a lot more of those strings together. And this is analogous to the fast twitch white muscle. So now, I hope that you enjoyed the lecture on muscular energetics, and thank you for your attention.